upper level.
You can. What? Oh my gosh, cold. You see it?
In.
Okay. Is the 30th. Okay. Paul, you've got it recording? Okay, good. All right, welcome back. We're going to pick up with genetics today and understanding inheritance of genes, and we'll talk about cell division and how that relates. What I want to do is start with a slide I'm going to share with you in a moment, and then I have a little Kahoot quiz to jog your memory from last time, and then we'll continue on with the Punnett Square stuff. So just so you all know, I'm going to upload a revised set of slides to include today, because I want you guys to know what some of the new terms are that you're going to learn that you'll need to know for the exam and for future units and topics that we're going to study. So you need to know what a gamete is, what chromosomes are, what we mean by heterozygous and homozygous, and then you'll also need to understand how these three laws that Mendel came up with based on his experiments relate to each of these terms on the right. So you will have this as your sort of vocab thing to go back to, so you can check to make sure that when you're taking notes, you're including the definitions for these terms. Okay, so let me see if I can get this Kahoot quiz to work. Yeah, here we go. We will do play. classic. There we go. All right, so let me share this so you can see it. Oh. I don't need that. Sorry if I blew your eardrums just then. I'll try to make sure I can default that off. So it's just a few questions, but you should see the pin for the game. So if you just go to kahoot.it and then enter in those numbers, either on your phone or I think your computer works fine as well, then you should be able to put in a little username and come right in. Do you guys see the?
It's showing your desktop, Dr. Jed.
Wrong. Okay. This desktop. There we go. Thank you. Should be a couple more people. Here we go. 20. All right, I'm going to hit start and we will talk about these. True breeding line or lineage works as well. Should all. Okay, good. So the yellow one is the correct one. Number 11, or excuse me, 11, the first one, the red one, I put in there because that was the sort of prevailing idea before Mendel came along, that you just get a blend of the parents. Like white bunnies and black bunnies would make you get gray furry bunnies, for example, kind of a paint blending method or model. But when Mendel selected those peas, he picked them because they were capable of selfing. You take pollen from a flower, put it on the same flower as a stigma, and get offspring, and they would look the same every generation after selfing. Okay, let's do the next one. Oh, we've got to do a little scoreboard first. All right, here we go. What are alleles? This one filled up quicker. Excellent, really glad to see that. Yeah, different versions of a gene, what we call alleles or alleles. Maybe a slight difference in the mutation will change the phenotype from flower color or eye color, or might influence height in a different way, but they all code for the same kind of a trait, like color or height or something. All right, new scoreboard. And last one, Mendel's law of independent assortment. I gave a little more time on this one. Aha, okay. So the first one is what we mean by independent assortment. You can think of that as every organism has 2 copies for each gene in the case of peas and humans. And exactly which one of the pair goes into each sex cell, into each gamete, happens independently of each other. So the genes that go into the sex cells for eye color have nothing to do with the genes that go into each cell for whether you can curl your tongue or not. And in the case of Mendel, the color of the P is sort of the gene for that, the allele for a color is divided up into the gametes independently of the allele for whether it's wrinkled or smooth, for example. Now, so the blue one, traits are determined by gene pairs, one is dominant, the other is recessive, is one of Mendel's laws, but it's not the law of independent assortment. And then the yellow one, the genes are separated such that each gamete gets one copy of each gene. That's his law of segregation. So the three of them are up there, but the one that I was looking for independent assortment is that the different genes for different traits are divided up independently of each other in the case of Mendel. I also put green here. Even though every individual has two copies, has a pair of alleles, the population could have more than just those two. So you could have an allele for blue eyes and an allele for green eyes, but somebody else has one for blue and one for brown, and they get brown eyes, for example. But in Mendel's simple model that we're going to talk about when we go back to the slideshow here, he only ever, stop share, he only ever had a two allele system.
Okay, for clarification.
Yeah.
So that last question, is that the same thing as what we talked about on Wednesday about how alleles are randomized?
Yes.
Okay.
Yeah. And the segregation is just that each sex cell only gets one of the two members of the pair. Stop share. Come back to PowerPoint. And okay. So just to recap, let me go to this slide real quick. The columns here, height, seed shape, seed color, seed coat color, pod shape, those are the different traits. And each of those has a single gene, but there's two alleles, a dominant allele and a recessive allele. So that's the organization of this chart. It could be that you could get a population of peas where the pods could be green or yellow or purple, but Mendel just didn't include any of the purple ones in there. So he only had two alleles in a system instead of three, even though each individual will only ever have two alleles. So let's go, before we get back into the Punnett squares, let's talk about something called ploidy level. In the case of humans, each individual typically has 23 pairs of chromosomes in a typical body cell, a somatic cell, like a skin cell or something. In the case of the peas, we're also dealing with an organism that has chromosomes in pairs. When you have two copies of each chromosome, you're called a diploid organism. And we usually abbreviate diploid as 2N, 2 times capital N. And that's why people say we have 23 pairs of chromosomes, or a total of 46 chromosomes. In this figure, what you're looking at are all the pairs of chromosomes. Notice that each chromosome is a single line with those red and yellow and dark stripes. When chromosomes replicate, that single line, that chromatid, becomes a replicated pair of sister chromatids that form a little X. So if you had replicated chromatids and you paired them up, each one of these, 1, 2, 3, 4, 5, each of these would have 4 lines. But these are unreplicated chromosomes that consist of a single chromatid. So that's why you get one and then its pair, one and then its pair. So at each spot along this chromosome, if you could zoom way in, you'd see a little stretch of DNA, and that would be a gene. And then that corresponding allele, the other gene of that allele, would be on the other side. If you only have one of each of these pairs, you're a haploid cell. You've got one N, or just N sets of chromosomes. Sperm cells and egg cells are haploid. Almost all of your body cells are diploid. You have a few unusual cells, like a red blood cell, where the nucleus is removed, and so you don't have any chromosomes in there. But for the most part, an individual cell in your body will have 23 pairs of chromosomes, unless it's a gamete. So here's a cartoon version of that. Here's a single chromosome comprised of one chromatid, and this band represents a locus, a little region of DNA, where you have the gene for flower color. And the spot where a gene occurs on a chromatid is called a locus. If there's more than one, you call it loci for plural. But if there's one, it's called a locus. So that's the locus of this gene for flower color. And there are two alleles in this example, an allele for white and an allele for purple. Make sense? And gene, of course, we already talked about what a gene is. Genes, you can think of them as particles of inheritance. They're your phenotype, the outward color of the flower in this case, is determined by what combination of alleles you get from your parents. There's two basic types of cell division in animals and plants that we want you guys to know about. And we have a link here to a nice YouTube video that explains the difference. Usually, when there's no cell division happening, inside the nucleus of a cell, you've got DNA, and it's kind of, you can think of it as kind of unzipped. It's not packaged into chromosomes. It's unzipped such that genes can be expressed. The enzymes that express the genes and transcribe the mRNA can get in there and do their work. When it's time to replicate the cell, you have to make sure that each daughter cell gets either a perfect copy of the genetic information or gets half of the genetic information in the case of making sex cells. Then all that DNA is condensed into chromosomes. That happens in a stage called prophase. So the formation of these chromosomes, shown here in the middle, once they condense, they're also replicated. So notice that this long blue chromosome here now consists of two sister chromatids united by the little black dots called the centromere. So this one is 2n equals 4. This is also 2n equals 4, except the chromatids are duplicated. After prophase, once the chromosomes are formed and the chromatids are replicated, these chromosomes then will line up along the middle of the cell called a metaphase plate, kind of a two-dimensional plane in the middle of the cell. Once they all line up, there are some spindle fibers that pull the chromosomes apart at the centromere and pull them towards each pole. So you end up with two identical daughter cells that look just like the parent cell. That's mitosis. That's typical, take one cell, make two copies. You start with a parent cell and you get two daughter cells and they're identical. This is happening all the time on the surface of your skin as your cells are being replicated. Meiosis is a highly modified version of cell division that results in the formation of gametes, of haploid gametes in animals. You can get meiosis that doesn't produce gametes, but that only happens in things like plants and fungi. In animals, when you have meiosis, the result are haploid gametes, or sex cells. Once again, the chromosomes condense and they replicate, but then notice that there are pairs of these replicated chromosomes. The long ones go together and the short ones go together. They form these tetrads. Tetrads are homologous chromosomes, and homologous just means they have the same size and similar, well, for this purposes, let's just say they're the same size, and there's always a pair of homologous chromosomes. In meiosis, when they line up on that metaphase plate, it's the tetrads that line up, not the individual chromosomes. Notice how in this drawing, the red ones are on the left and the blue ones are on the right. That can happen in any order. You could have the reds on the right and the blues on the left, or you could have blue on the right and red on the left in one, and red on the left and blue on the right in the other. That sort of shuffling of just lining up all those tetrads in the middle, that is Mendel's law of independent assortment. He couldn't see this. He didn't have a microscope. He didn't know what the cells looked like. But the mathematics that he used to predict how inheritance happens actually plays out right here as the tetrads align randomly on the metaphase plate. Because which side you end up on, left or the right, determines which of these two daughter cells of meiosis 1 the chromosome ends up in. That's a critical point. Once you're in this, at the end of metaphase 1, you have two daughter cells. Then the chromosomes that are in those daughter cells line up the metaphase plate again, this time kind of like in mitosis, and the two sister chromatids are pulled apart until you end up with a total of 4 daughter cells, each of which are haploid. So it's in meiosis 2 where you really see Mendel's law of segregation, where each gamete gets one copy of a gene, not both. But it's in meiosis 1 where you get the law of independent assortment, where the tetrads shuffle up. This is challenging, and that's why we provided the video with you as well, for you as well. But metaphase 1, this is where independent assortment is, and meiosis 2 is more or less where segregation happens. Okay. So where's my mouse? Quick review, you saw this slide already. Key features of single gene inheritance. The genes come in pairs for each individual. For each pair, the alleles might be the same or different. So you saw an example with a purple flower color and a white flower color allele. You could have 2 for purple or 2 for white. That's fine. Depends on the individual. Each sex cell gets only 1 allele. That's what we saw at the end of meiosis 2 with segregation. And then when you put 2 gametes together, you reunite the allele pairs and you get a diploid zygote, or a diploid individual can develop. So we talked about Mendel's true breeding lines, big R, and little r. Those are homozygous. One is homozygous dominant and one is homozygous recessive. And we typically use the lowercase for recessive and the uppercase for dominant. Separate those out, figure out what the possible genotype is of the gametes. When you're homozygous, it's pretty easy to do that because there are only 2 possible and they're the same. But then you can bring those together and you get a next generation. In this case, you will always get a big R from one parent, a little r from the other parent, no matter which gamete you get. But if you take that heterozygous individual, meaning one dominant and one recessive, and you cross it with another similarly heterozygous individual, you set up a Punnett square, where you identify the gametes from one parent on the columns and the other parent on the rows. Then reunite them, because you're bringing those 2 gametes together, an egg cell and a sperm cell together, putting those 2 alleles back together so you have a complete pair. And there are 4 possible outcomes. Of those 4 possible outcomes, there are actually only 3 possible genotypes, right? Big R, little r. And how many possible phenotypes are there?
2.
Yeah, good. Okay, so the next question is, what if you're interested in more than one trait, not just round or wrinkled, but you want to predict how many of your offspring are going to be yellow and wrinkled, for example? You can do that by tracking multiple alleles. So the genotype for this yellow P, for the 2 alleles of interest, remember there are many genes in the genome of this P, but we're only interested in 2. In a true breeding line is big R, big Y, big Y. So 2 genes for roundness and 2 genes for color, yellowness. And then we're going to cross that with the opposite, a green and wrinkled one. The trick here is figuring out how to make the gametes. Each gamete gets 1 copy of each allele. Can someone predict what the gametes are going to look like for the yellow round seed? A gamete has to get 1 copy of each of the 2 genes.
Would it be big R, big Y on the top and little r, little y on the bottom?
That's correct. Thanks, Joe. So you have to pick an allele from the first pair of genes and then pick an allele from the second pair of genes. In this case, because they're both homozygous for both genes, there's only 1 possible gamete in each case.
What would happen if it was heterozygous?
You would have more than 1 possible gamete produced. So you'll see that in the next moment, actually. So if you breed this round yellow plant with the wrinkled green plant, unite those 2 gametes, then you get a parent that is exactly what you just said, Connor. It's heterozygous for both. It's got a big R, and a big Y, little y. Now, Mendel took these, which are also yellow and round, and crossed them. Can someone remind me which of Mendel's laws explains why, if you have a plant with a genotype big R, big Y, little y, how come it looks the same as the round yellow parent? Why is that phenotype the same? Which law tells you the answer to that question?
Law of dominance?
Yeah, good. So if we take these, what are the possible gametes that you could make here? How many different ways could you put R's and Y's together to make a gamete? You guys remember ever having to use the FOIL method in a math class?
Yes.
Yeah, right. First, outside, inside, last. You can use that here. So let's start with the first, big R, big Y. First allele from this gene, first allele from this gene, big R, big Y. Outside, big R, little y. Inside, little r, big Y. Last, little r, little y. That always works. But if you did the same thing with the parents, the big R, big Y, you get the same one every time, right? But keep that FOIL method in the back of your mind, because you can always use that to figure out how many gametes there are if you cross heterozygous individuals for 2 genes. So we call this a dihybrid cross. The di means 2, because you've got 2 genes. You're crossing 2 individuals. And in plants, anytime you cross 2 plants, we call that a hybridization event. In animals, usually we think about hybrids are 2 different species, like a donkey and a horse, right? Hybrid's a mule. With plants, we pretty much just always call it a hybrid if you are forcing the cross between 2 individuals. So you array the 4 possible gametes along the columns and the 4 possible gametes along the rows, and you get a 4x4 grid. Put them together. What's the genotype of this top square going to be right up here?
Big R, big Y, big R, big Y?
That's correct, but we tend to keep the alleles that go together.
So big R, big R, big Y, big Y?
Exactly. Yep. And so you do that for each one of these, and you get all the genotypes. Then you have to think about Mendel's law of dominance to figure out what the phenotype is going to be for each of those. Wrinkled and round, wrinkled and yellow, or wrinkled and green, round and green, or round and yellow. What's the ratio of green and wrinkled to the rest of them? What proportion of the offspring are green and wrinkled?
1 to 16.
Yeah, 116.
Yep. So you can think about these ratios. I could also ask you, what's the ratio of green to yellow?
One-fourth.
Yeah. So see, first time I asked about a pair of genes for the phenotype, and the other case I asked about just one of the two alleles. So this is what you get, right? 9 yellow, 3 yellow and round, 3 wrinkled and yellow, 1 green and wrinkled, and 3 green and round. This is all possible combinations. You could take any two of these offspring and cross them again. Remember, these are ratios because you could do a cross like this and you had to plant a lot of bean pods on it. You could have hundreds of peas, but they would generally be in this ratio. And that's exactly what Mendel found. In his F2 generation, he got 556 seeds, and these are the actual numbers of the different types of peas that he got. And when he looked at the ratio, 9.84 to 3.16 to 3.36 to 1 out of 556 seeds. So you can compare that back to the 9 to 3 to 3 to 1 that's predicted here at the bottom of the square. If you don't get these ratios, it's because something is preventing the system from following Mendel's laws. And I keep putting laws in air quotes because they're patterns that he found and rules that are sometimes followed, but certain things can happen to make them not followed. For example, each of these genes is actually on a different chromosome. And that's part of why this all works. Each gene is on a different chromosome. We already kind of covered this. Each trait is a single gene with 2 alleles. Could have been more, but Mendel chose to only focus on ones with 2. And the probability of getting any one of the 2 alleles is half. They're randomly assorted at metaphase 1. Let's see what I had here. We kind of talked about this already, about how many different genotypes, how many different phenotypes, but let me ask you guys about this for the dihybrid cross again. In the resulting square here on the right, how many different phenotypes do you see, considering the 2 traits?
4.
Yeah, good. And how many different genotypes are there?
Eight, maybe?
Yeah, 1, 2, 3, 4, there's a pair, 5, 6, 7, 8, 9. So there's a symmetry here that you can use to kind of understand how the square works. And as I already said, again, this is a cross where the 2 parents have the same heterozygous genotype at both loci. But you could take any 2 of these from the square and cross them, and you would get some smaller number of genotypes and phenotypes. Now, the reason I talked about Mendel's laws in quotes again is that there are situations where you don't necessarily follow, where things don't necessarily work the way he predicted. One of those conditions is called incomplete dominance. So here's a case where the big R is an allele for the red color and the little r is an allele for white color. But if you're heterozygous, you're not just red, you're pink. So it is kind of intermediate. It does kind of seem like blending. But if you cross the pink ones together, you can get red ones and white ones back the next generation. And that doesn't work in a blending world. It only works in a particulate world. You can see that result here, right? If you take two pinks, you should get how many, what proportion should be red? So is it on there? Let me ask the question, how many genotypes do you get if you cross two pinks?
Three.
Yeah, 3, right? And how many phenotypes do you get?
3.
Yeah, if it was complete dominance, you'd only get 2. Good. Sometimes this situation can arise when the amount of pigment that's produced depends on the number of genes involved. In this case, there's only one gene, so you either have half the amount to get red, so you're pink, or you have the full amount to get red, so you're totally red. Some traits are coded for by many genes. So you could have several different genes that all contribute to the production of red pigment. And so the number of dominant ones you have versus the number of recessive ones you have at these different genes will affect sort of how red or how pink the resulting flower is. We call that dosage effects. So if you had a situation where there were 3 different genes, each of which has a pair of alleles, and if you have dominant ones, you're producing that red pigment, and if you have recessive ones, you're not producing any red pigment, so you're not as red, then you can actually do a cross where you figure out, well, each gamete has to have 1 copy of each of the 3 genes. So the gamete gets the genotype ABC, it can be capital or lowercase. There's a lot of possible gametes that you could make there, right? They're all arrayed here across the top. And if you cross something like this with the same kind of plant, so you got the same possible genotypes for the gametes, can someone tell me what the genotype would be of this offspring here in the lower right corner?
Little a, little a, little b, little b, little c, little c.
Yeah, so it'll be homozygous for all 3 genes, all 3 loci, and you can find combinations where you'd be heterozygous for all 3. So you can fill that in. How many different possible combinations are there? How big is this table? Yeah, 64 squares, right? Now, this is getting a little bit unwieldy, right? It's a lot to write. But if we think about the dosage effect for a moment, and we think if you've got a dominant allele that's going to contribute to a red color for the flower, or for this example actually comes from red wheat, I think. But if the dominant is for redness and the recessive doesn't contribute to any redness, then what you can do is just count up how many dominant alleles you have. So in this second one here, how many dominant alleles are there? First row, second column.
5.
Yeah, 5. Good. Down here in the middle, notice we got 3 out of 6. You can just plug in all those numbers, and you can once again see that symmetry that we talked about before. 6 in the upper left, 0 in the lower right, and somewhere in between everywhere else. We can make that visual by providing a color that matches the dosage of those dominant alleles. And you get a square like this. So most of them are going to be in this pink zone here, 1, 9, 9, and 1. Some of them are going to be a little redder, some of them are going to be really red, and a few of them are going to be really very white. And so you get what looks like continuous variation. Lots of traits are like this, where there are many different genes. It's a polygenic trait, many genes. And so in the population, what looks like kind of blending, you mix red and you mix white and you're going to get a bunch of pink offspring of various shades. That looks kind of like a blending world, but it's underlain by a truly particulate inheritance model that we know about because of Mendel's experiments. And if you look at if you were to produce, say you're a wheat farmer, right? You make this cross and you produce dozens or hundreds or thousands of seeds. You could then plot the frequency of each color. Or if you're breeding snapdragons like this, you could plot the frequency of the different colors, and you would end up with a graph that looks like this. If you have a lot of dominant alleles, you're over here on the right. If you have very few dominant alleles, you're over here on the left. If you've got an intermediate, you're here. And this is the frequency of offspring with some number of dominant alleles or doses. And you get mostly pink offspring. Now that only works if you're crossing two things that were heterozygous for every position, right? But this is how we can link Mendel's particulate inheritance to the real world that we see around us all the time of continuous trait variation. For example, height, or even eye color isn't actually just blue, green, or brown. There's lots of intermediates, right? This is kind of more typical, but it's underlain by genes as little particles of inheritance. Okay, that is where I have it. Have us stopped for the basics of inheritance. Stop sharing here. Are there any questions about these Punnett squares, or about what we just talked about? Or any questions about the vocab words at the beginning? Oh, actually, one that, since no one's piped up yet, one thing that I will point out that I'm not sure how clear I was last time with the vocab is this word polymorphism. Even though each individual only has 2 copies of a gene, because it's diploid, there might be more than 2 alleles in the population. If that's the case, the population is polymorphic for that gene. That's different than what we just talked about, where you have multiple different genes contributing to the same trait. This is about how many alleles are there for a given gene or locus in the whole population.
So Nathan, you ready for me?
Yep, if there's no questions, I'll hand it over to Paul.
All right, I'm going to share my screen. Dr. Judd's already talked about a little bit of what I'm going to talk about because he's bridging the gap between these simple traits that are governed by single genes that there's an either or sort of inheritance, what we call particulate inheritance is what he's referred to. So I'm going to actually be talking about more about these polygenic traits. So can you guys see a cat on your screen?
Yes.
Everybody see it? All right, cool. Okay, so these quantitative genetic traits or additive genetic traits get analyzed in different ways than we analyze traits that are demonstrating what we call particulate inheritance. And so I'm going to throw a number of things at you. I'm going to revisit a simple monohybrid cross, but I'm going to throw some math into it that will help you see how selection can act on a trait. And then we'll use those ideas to bridge this gap into quantitative or additive genetics. So one of the things that we're going to continue to work on is discussing how these additive genetic variants of polygenic traits lead to continuous variation in phenotype. That's what Dr. Judd just went over with you, and we'll revisit that. I'm going to introduce you to an idea called Hardy-Weinberg equilibrium, what it is, how it works, and we're going to look at how we can apply Hardy-Weinberg equilibrium to understand how selection works. And we're going to use an example of Manx cats to do this. And then ultimately, we're going to look at how we can determine the heritability of quantitative traits and use the breeder's equation to predict evolution from one generation to the next under the breeder's equation. And we'll learn how to estimate heritabilities using regression. So for those of you who have not been in class, in lab yet this week, tomorrow Dr. Judd will go through regression with you. I also kept the recording of lab yesterday when I was on with my students. So there is an 1 hour and 52 minutes recording on the YouTube site that you can also go and look at. If you have an 1 hour and 42 minutes or an 1 hour and 52 minutes of patience to get through it. So Dr. Judd was just showing you this graphic where we have a trait underlain by 3 genes that gametes for that will produce a F1 generation. Dr. Judd, I don't recall, did you introduce terminology like F1 and parental generation and all that?
Actually, I didn't explicitly say that. I use those terms and they're on the slides, but I don't think I paused to explain those terms.
Whenever we do a genetic cross, this first generation, in most cases, it's a pure breeding or true breeding individuals, homozygous for all the traits. This would be the parental generation. The parental generation creates gametes. I always circle my gametes just to indicate to myself that they're gametes rather than genotypes. But we should also know that they're gametes because they're not diploid, they're haploid. They only have one representative of each gene. And then this first generation that comes out of that cross is generally referred to as the F1. F1 stands for the first filial generation. And we generally shorthand this F1. And so in this case, it gives you these heterozygotes. Dr. Judd took you through this cross. If we let those heterozygotes breed with one another, we get this horrendous mess of a Punnett square, 64 items big. And what a quantitative trait basically does is it says, for each dominant allele, you get something added to the phenotype. In the case that he was talking about, you get pigment added to the phenotype, but it could be anything. This is actually the way height works. This is the way sprint speed works. This is the way a great many traits work. And so you end up with this figure that he has already shown you. And so these quantitative traits or additive traits, you can call them either quantitative genetic traits or additive genetic traits. I will flip back and forth between those two terms quite a lot. I recommend that maybe you learn it as additive traits because that's most descriptive and kind of how it works, and that the dominant alleles are adding to the phenotype. But both of those terms mean the same thing. The reason that they're called additive is because of the action of the dominant alleles. The reason that they're called quantitative is because we use quantitative methods for studying their inheritance rather than some other set of methods. So we use math to study quantitative genetics, but remember that that math is rooted in simple Mendelian processes that are applied to multiple genes. So now what I want to do is go back to a simple monohybrid cross. So we have individuals that are homozygous dominant and homozygous recessive. We create gametes from those individuals. We get a heterozygous that is in the F1. If we allow those heterozygotes to mate with one another, we will then generate your standard Punnett square, and we will get these genotypes: a homozygous dominant, two homozygous recessives, two heterozygotes, and one homozygous recessive. 3 of these individuals will have one phenotype, 1 of these individuals will have another phenotype, if it's one of these traits that is particulate in nature in terms of its inheritance. If it's not, if it's an additive trait, 1 of these individuals will have one phenotype, another 2 of these individuals will have a phenotype that is not quite as strong as the first one, and then the last individual will have a phenotype that essentially lacks that trait, whatever the expression of that trait is. Well, we can think about these different alleles as having a frequency in the population. So if we have a population of only 2 individuals, 50% of the alleles in this population are the big A allele, the dominant allele, and 50% of these alleles are the little a allele. In a similar fashion, if we have a homozygous dominant and a homozygous recessive individual, there are once again 4 alleles in this population. 2/4 of those alleles are dominant and 2/4 of those alleles are recessive. And so we can refer to the frequency of these alleles using numbers. So the frequency of the dominant allele in this case, we will generally refer to as P, and the frequency of the recessive allele, we will generally refer to it as Q. And in this case, the frequency of P is 0.5 and the frequency of Q is 0.5 because half of the alleles in the population are the dominant allele and half of the alleles in the population are the recessive allele. Now, if we go to the F2 generation, which is this generation, once again, we have 4 different kinds of offspring. If we have 4 different kinds of offspring, 1/4 of them are homozygous dominant, 2/4 of them are heterozygous, 1/4 is homozygous recessive. If we have 4 individuals, we have 8 alleles. 1, 2, 3, 4 of those alleles is the dominant allele, and 1, 2, 3, 4 of those alleles is the recessive allele. So once again, we have 4 8s and 4 8s. So we're still at a 50-50 mixture of these alleles. Now, here's where things begin to get complicated. We have 1/4 of the population that is this genotype. We have 2/4 of the population that is this genotype, and we have 1/4 of the population that is this genotype. Who wants to try and figure out how to predict what will be the mixture of genotypes in the next generation? Using the tools that you have before you.
Would we make a Punnett square with all 3 of them?
Oh my gosh, how would we do that? So we could basically say, okay, well, these individuals can make only 1 type of gamete. These individuals can make 2 types of gametes. These individuals can make 1 type of gamete. So that's a start. But now you're talking about a 3-dimensional Punnett square, aren't you?
Yeah.
But not only, what's that?
I was just agreeing.
A 3-dimensional Punnett square, you would need one. So if we were doing this time, this is a 2-dimensional Punnett square. But if we were to add a 3rd type of individual, we would have to make a Punnett square that went out this direction. And boy, doesn't that become complicated now? Because along this axis, and it would have to have, it would end up being a 3-dimensional Punnett square because we would have to have the third genotype type. Not only that, there are way more of these individuals, so way more of these gametes than either of these gametes. because there's only 1/4 of the population that is this and 1/4 of the population that is that. What's a person to do? Well, what we do is we abandon Punnett squares. Punnett squares are useful for what they're useful for, and then they're totally not useful for what they're not useful for. But then again, couldn't that be said about just about anything? What we can do is, in this population, we have 2/4 of the population that is this, 1/4 of the population is this, 1/4 of the population is this, 4 individuals with 8 alleles. 4/8 of those alleles are the big A allele, 4/8 of those individuals are the little a allele. As it turns out, for any frequency of alleles, we can figure out what the mixture of genotypes will be in a randomly breeding population. And we do it by using these allele frequencies. So if we replace this Punnett square with the allele frequencies instead, we can figure out, well, what's the frequency of the homozygous dominant rather? What's the frequency of the heterozygotes, and what is the frequency of the homozygous recessives? P times P is P squared. You have PQ for the heterozygotes, but there's 2 of those represented in the Punnett square, and Q squared is the frequency of the homozygous recessive. So these are now genotype frequencies. And I want to caution you, be careful when you're using terms, and we'll do our best to be careful when we're using terms. There are allele frequencies, which is what I just erased up there, and then there are genotype frequencies. So the Hardy-Weinberg equilibrium equation allows us to compute genotype frequencies if we know allele frequencies. So P and Q are allele frequencies. P squared to PQ and Q squared are genotype frequencies. So you'll notice that we started out with allele frequencies of 0.5. We got an F1 generation that had an allele frequency for each allele of 0.5. We allowed those individuals to breed, and they produced a set of offspring that had an allele frequency of 0.5. If we were to allow all of those individuals to mate with one another, we could figure out what their allele frequencies would be using the Hardy-Weinberg equilibrium equation. And so we can just take this mathematical formula and say, well, what's the allele frequency of these offspring? It's 0.5 squared, 2 times, I'm going to actually do this with fractions instead, times 1/2 times 1/2, and this would be 1/2 squared also. So this would tell us what the genotype frequencies are. So the genotype frequencies would be 1/4. The genotype frequencies here would be 2/4, and the genotype frequencies here would once again be 1/4. It's like having 1 individual of this, 2 individuals of this, and 1 individual of this genotype. Once again, we have 4 of the 8 alleles are dominant, and 4 of the 8 alleles are recessive. The allele frequency doesn't change from generation to generation. So when you hear somebody talk about Hardy-Weinberg equilibrium, the term equilibrium just basically means that it's not changing. You'll get the same allele frequencies generation after generation after generation after generation, unless something happens.
If I can,
Yeah, go
if I
ahead.
can pipe in here, notice that if you take that 1/4, 2/4, and 1/4, those are the proportions of the genotype frequencies in the population. What do you get if you add them all together?
I forgot to add the equal part.
Anybody? What's the sum of those?
This is the easiest fractional math in the world.
1.
It
One.
adds up to 1. So 4/4 is 1. So this thing should always add up to 1.
Because that's everybody in the population. In this case, the alleles are divided up among the genotypes randomly. That's what we mean by equilibrium. If they were not apportioned to the genotypes equally, such that you had a different set of 3 proportions, but they still add up to 1, you would not be at Hardy-Weinberg equilibrium. But no matter whether you're at equilibrium or not, it's always going to equal 1 because you're always counting everybody. The same is true if you add P and Q to look at the allele frequencies instead of the genotype frequencies. All of the alleles sum to 1.
Yes, that's an important point. So at equilibrium, allele frequencies do not change, and as a result of the allele frequencies not changing, the genotype frequencies will also not change over time in this particular situation. So let's write down some conditions under which Hardy-Weinberg equilibrium will hold. So what Hardy-Weinberg equilibrium basically says is that under certain conditions, allele frequencies do not change over time. What are some of the conditions under which this might happen? I don't think this is in your textbook. We haven't covered this in class. I'm asking you just to think. What's one situation that we've talked about in which allele frequencies will change? We've talked about it a lot so far.
Natural selection.
Okay, good. I was about to pull up an example. So what's going on with natural selection? Imagine that you have a population of horned lizards, and we look at horn size, horn length, and if this is one of these traits that is underlain by lots of different genes, each one contributing to horn length, what would you expect the shape of this phenotypic distribution to look like? What shape distribution do we get out of these quantitative traits?
Assuming there's no shrikes to worry about.
Assuming that there's no stripes to worry about. You guys can still see my screen, right? What shape is that?
Are you looking for normal distribution?
Oh my God, Grant, I'm looking for a normal distribution. Thank you very much. So In this normal distribution, there are some individuals with really long horns, there's some individuals with really short horns, and there's a ton of people here, a ton of horned lizards here in the middle that have kind of medium length horns. Now, in the case of the shrikes, what's happening to this population as shrikes prey upon them? Somebody other than Grant. Who gets selected against in this system?
The shorter horned lizards.
So there's selection against shorter horned lizards. And because of this, because the shortest horned lizards have a genotype for horn length that is something like this, these guys have a genotype for horn length that is something like this, these individuals have a genotype that is something like this, or this, or something like that. We're losing more of the recessive alleles when selection acts on this population. And as a result, the allele frequency changes in the population. So certainly one of the things that happens is that there's no natural selection. So if there's no natural selection, we should expect the population to have the same allele frequencies from generation to generation. There are about 8 assumptions for Harding-Weinberg equilibrium to stay at equilibrium. We'll teach you the 4 most common ones that are mentioned. Between now and Friday, make an effort to go and look those up, and we'll continue with this discussion on Friday. Also, between now and Friday, take your conversation pills, because I'm going to be asking you a bunch of questions and I need you to give me answers. Otherwise, we have these awkward pauses that Grant has to save us from. Thank you, Grant. So we're going to cut off that for today. There is one thing that I want to talk to you about before I let you go, though. We gave back your tests on Monday. We sent them out to you. And there was some concern expressed to us, to me, and I don't know if this was also expressed to Dr. Judd, that there was some academic tomfoolery going on during the exam. We have no way of knowing what you're doing while you're taking the exam, but I will say this: if people were seeking out help from other people during the exam, you need to find different people to reach out to, because here's a figure of the results from last year's exam versus this year's exam. And why is this so gigantic on my screen?
That looks a little better.
Yeah. So, whoa, where did it go? Ah, here it is. So basically, the exam one from last year was 69.5, and this year it was a 69.22. And these are the 95% confidence intervals for the 2 exams. So what we're going to do next exam is we're going to require that you all have your cameras on and you all have your microphones unmuted, just to add a little bit of security. But this graph shows that if you are seeking out help from people, you need to find a better class of people to seek help from, because they're not helping you. One of my fears in giving the first exam, Grant's laughing. Grant, are you laughing? One of my fears was that we would get the exams back and the average would be 77 or something like that. But the fact of the matter is you guys performed exactly like every other first year class has performed. Part of that is that we teach you in a different way that you probably weren't taught in high school. And the other thing is that we're probably testing you in ways that you weren't tested in high school. And it takes you a little while to get used to that. Generally, we see test scores go up over the course of the semester because you're adjusting to both how we're teaching and you're also adjusting to how we're testing. One of the reasons we gave you an old test to look at was so that you could be a little more adjusted to that than you normally would be. But as you go through the semester, you will get better at taking the types of tests that we give you. So we'll remind you of test taking procedures as we get close to the next test, but this is the result for this test. So it's not stellar, average for an exam in college. We would like to see better exam scores, but just know that you're not really performing above or below any other class that we've had of first-year students. The first exam is always a little rougher than the others. Questions about that? All right, we'll see you on Friday. Look up the things about Harding-Weinberg equilibrium. Under what conditions do we stay at equilibrium? And we'll cover that at the beginning. Dr. Judd, you're in charge, but I think I have the recording, so I'll upload it.
Yeah.
I don't know if you had a chance to look at my protocol. It's freaking long.
Yeah. I
And the other,
I previewed it and then I modeled my edits on it a little bit, so it's similar format.
okay.
But I'm going to look a little more carefully after the chair's meeting this afternoon.
Yeah, okay. I'm not sure how much of the safranin that they should be adding, so I'm probably going to alter those amounts. And the other thing is that dropper that you had that it came with, that seems very imprecise. So
Yes.
I think what we should have students do is use a pipette to do that.
Yes, for yours especially.
Yeah.
For the pollen load, they should use a pipette. I think that stain is strong enough, and I don't think it needs to be more concentrated.
Oh no, if anything, it needs to be more dilute.
Yeah, yeah.
Because one of my worries with the stigmas is that it also stains the stigma, and if there's too much in there, it's going to stain it so that you can't really see through things very well.
Well, it should only stain tracheids. It shouldn't stain the regular parenchyma cells.
Well, why did the stigmas look so red when
because of the camera
micrograph?
because of the camera through the microscope.
Okay, all right.
Because I was holding the iPad up.
Yeah, so I need to go and get some bugs and vortex them, and then do a couple of tests with the safranin. Where is the safranin? Is it in your office or is it upstairs?
It's in the fume hood in the E&E lab.
Okay, so I'll do some tests and kind of decide on what an amount is to put into the pollen load tubes. So.
Yeah. I have more stain. I think we have plenty, but I have more of the powder in my wet lab, and also there's a bunch of it in the cabinets over by Dr. Reynolds' lab.
Okay, all right.
The lab place. There's more there too.
Well, good. I'm not really worried about running out of it. I just was like, why use more than we need to? Right. And for those guys, they're just looking at it against a light background. All it needs to do is stand out and be visible.
They should pop.
So, yeah, certainly. All right, I'll talk to you later.
All right, see ya.